Okay, our last little video clip here for Chapter 2, we're going to talk a little bit about preparing specimens for microscopy and staining. And I'm not going to get too much into this. I just want to hit some highlights on it because you're going to have a whole Learn Smart Labs activity on bacterial staining, which we'll go into a lot more detail on this topic. All right, let's say you have a petri dish with colonies of bacteria on it that you have cultured, you've grown them in the laboratory, and now you want to look at the individual cells, those bacterial cells under the microscope. So typically what you do in the laboratory to start off with is called a bacterial smear. So you take some of your sample and you smear it across a glass slide and you let it dry. You have to let it air dry. If you were starting with, now here you're starting with a liquid culture, but if you were starting with a colony on a dish, you would have a little drop of water on the slide, put your bacterial cells in there, smear it out across the glass slide, and let it air dry. And then you do what's called a heat fix, where you actually take your slide and pass it through the flame. And two things, that makes the bacteria stick to the slide, and secondly, it kills them because you don't want to, you might be working with some sort of bacteria that are potentially harmful or pathogenic, and so you want to make sure they're dead on the slide. That's called fixing the bacteria to the slide, or heat fixing. And then finally, you apply some sort of dye, some sort of stain that will attach to the bacterial cells but will not attach to the glass slide. And that is going to improve something called our contrast. You're going to improve the contrast. Cells typically themselves are colorless, but when you apply a stain to the cells, they take up that stain, that dye, and now you can see them better against that clear background on the slide. And finally, you have to rinse your slide. You have to get rid of that excess dye, wash it off the glass so that you can see your stained cells and they have better contrast against that lighter background. All right, these dyes that are typically used, there are different ones that can be used in the laboratory. One example is crystal violet, which has a dark purple color, and another one is called methylene blue. Those are both very commonly used in microbiology laboratories. And one key thing about these dyes, many of the dyes that are used to stain cells are positively charged dyes. So they have a positive electrical charge. Cells, as you guys learn about in anatomy and physiology, tend to have an overall negative charge. So if you total up all of the charges inside cells or around the cell membrane, you have an overall negative charge. So positively charged dyes are very strongly attracted to cells. They bond very tightly. Unfortunately, if you're actually doing this in a real lab and not a virtual lab, they also love to stick to your skin cells. So you tend to get these dyes all over your fingers if you're not wearing gloves. But of course, you're always wearing gloves in the laboratory, right? That's part of our safety training. But anyway, they're showing you here one type of stain that's commonly done in a microbiology laboratory is a simple stain, and that's not really given that name because it's easy to do. It is easy to do, but simple means you're using one type of dye. And so here are some E. coli, Escherichia coli cells stained with crystal violet, and here are some Corynebacterium bacterial cells that are stained with methylene blue. That's a simple stain, one dye. Differential stains typically use two dyes, and different cells, different species will come out different colors based on the types of dyes that are being used and your staining process. So again, this is called a differential stain. It's going to help you differentiate or distinguish between different types of organisms based on the color they come out in this staining process. So one of those differential stains is called the Gram stain. So the Gram stain uses a purple dye called crystal violet. It uses a pink or reddish dye called safranin, and bacteria tend to either come out purple or pink. Purple cells are referred to as gram-positive, pink cells gram-negative. The positive and negative there does not refer to the electrical charge. It's just referring to the result of the Gram stain. Purple's gram-positive, pink gram-negative. So you can see there in the picture, you've got a mixture of bacterial cells in there of different sizes and shapes. some are purple, and some are pink. You're going to have a whole activity where you do gram staining as part of a virtual laboratory at the end of this unit. So you'll hear a lot more about the gram stain. And as we get into Chapter 3, a lot more about the gram stain as well. This is your most commonly done staining process in the microbiology laboratory. The acid-fast stain is another type of 2-dye stain that's done. You get red cells and blue cells, but for different reasons than you do with the gram stain. So red cells are considered to be acid-fast, and blue cells are non-acid-fast. This is a type of staining process that's typically done to try to detect tuberculosis bacteria. They are the tuberculosis bacteria come out red in an acid-fast stain. Most other types of bacteria come out blue. And here's another one called the spore stain. So toward the end of Chapter 3, we're going to learn about how some bacteria make a dormant structure called an endospore. And you can actually see these spores pretty well if you do a process called a spore stain. So again, you use 2 dyes, and the spores, these dormant, hardy, protected structures that some bacteria can make, come out red. And the vegetative cells, vegetative there means active live cell, not like a cell that's in a coma or something like that. Vegetative means live and active. Endospores are dormant, vegetative cells are active. And so when you do that type of process, those vegetative cells come out blue. A lot of people use a green dye for the vegetative cells when they do an endospore stain. All right, so just wanted to hit just a couple of highlights there on staining. Again, you're going to get a lot more of that information as you go through your Learn Smart Labs activity. So that's going to wrap up the highlights we needed to cover for Chapter 2. So be sure you finish up your homework and quiz activities. And ideally, if I were you, I would go ahead and take that Chapter 1-2 exam, review everything, and go ahead and move on if it works with your time schedule, and or go on and begin your Chapter 3 activities.